Okay, for this week, we're going to be talking about IgE and some of its receptors. So this is really important because IgE is one of the main ways that mast cells are activated during allergic reactions. And IgE is actually at the core of a lot of atopic conditions. And so last week we talked about mast cells, and this week we're going to be talking about IgE, and they go quite hand in hand. So just to give an overview, IgE is, of course, an antibody, so we'll talk about its structure, we'll talk about how it's synthesized, and we'll talk about its role in allergic signaling, and we're also going to talk about its main receptor and its secondary receptor as well. So to give an overview, as I said, IgE is sort of at the core of a lot of allergic diseases, and it's different than a lot of the other immunoglobulins because it acts much, much faster, and it can sort of amplify a very small allergen signal into a very widespread and diffuse response, which is why even the smallest amounts of, like a peanut, for example, could launch someone into a systemic anaphylactic reaction. But it's also important to know that it's not just a hypersensitivity protein. It has a lot of roles in innate immune suppression, responding to respiratory viruses, handling T cells and immunosuppressive T regulatory cells as well, parasitic immunity. So again, it's this through line of things that seem sort of detrimental at first, but if you take a much more bird's eye view, they actually have a lot of functions that might have been evolutionarily advantageous as well. So talking about the structure of IgE. Now, human IgE is very rarely found just floating around in your bloodstream. So almost exclusively in humans, it's found bound to its high affinity receptor, which is this Fc epsilon R1 receptor. And it could be bound on these receptors for an extremely long amount of time, up to 2 months. So the structure of the actual IgE antibody itself is a tetramer. So it has 2 heavy chains and it has 2 light chains. Now, if we dive a little bit deeper, we'll see that the heavy chain has this N-terminal variable region. And that's the region that creates antigen specificity. And it's called a variable region because it is not the same on every single IgE molecule. It's obviously different, and that's why it is able to be specific for certain antigens or allergens. Now, the constant regions, on the other hand, on the C-terminal of the heavy chain, are going to be conferring the more isotype-specific functions, such as receptor binding. And if we look at this C epsilon 2, and C epsilon denotes the heavy chain, this is a hinge region. So if you can see on the left side of this image here, that's the hinge. And then the C epsilon 3 is containing the glycan, the sugar that's actually necessary to bind to the high affinity receptor. So a lot of times you might have seen IgE or antibodies displayed as a sort of like very rigid Y shape, but it's a little more intricate than that. So this image here is a really good representation of how which parts are doing which. So there's the hinge region, there's a variable region, constant region, and then also the actual binding region. So obviously it's made by B cells, as all antibodies are. It's secreted by plasma cells. And there's actually a very low amount of IgE positive B cells because typically they're going to more rapidly transition to the plasma cells that are secreting IgE into the bloodstream. So as far as a general overview, I have this image here. There's a lot of steps, but what I want to highlight is that this follows a very similar formula to what we've seen. So antigen-presenting cells, such as the dendritic cell, are going to activate the T helper 2 cells. These cells are going to engage their B cell counterparts with various co-stimulatory signals. That's going to begin germline transcription of the heavy chain locus at for the constant region, and it's going to begin expression of the AIDA enzyme, which is activation-induced cytidine deaminase. So let's just dive into a little bit of this image here and go over some key steps. So if I had to sort of distill this complicated image into just a few steps, I would say that the first thing is going to be the antigen binding, and that is going to be processed into a peptide presented onto an MHC class 2 molecule presented to a T cell. The T cell receptor, or TCR, is going to engage. You're going to have all of these sort of co-stimulatory signals, and then you're going to get some cytokines that are going to be produced. So those cytokines, like IL-4 and IL-13, are going to promote the isotype switching and activate transcription factors such as STAT6, and STAT6 are going to begin the transcription of the IgE heavy chain. And then also this CD40 molecule that is going to get expressed upon antigen exposure is also going to be relevant in signaling the induction of the AID enzyme, which is useful for DNA recombination, which gives us the class switching. So really the big takeaway here is that you have just a lot of interaction, and this is sort of how we can visualize antigens connecting to T cells, connecting to the cytokines, connecting to the transcription factors, connecting to the actual transcription of the IgE heavy chain. So hopefully this provides a rough idea of how things are related, but again, things are just very interrelated, and everything that we've been learning about so far is often related to other things within the realm of allergy and atopic conditions. So let's talk about the high affinity receptor. The high affinity receptor is going to be the main way in which IgE interacts with antigens. So if an antigen ever comes into contact with an IgE, the chances are it's actually already loaded onto the high affinity receptor. So there's two dominant forms. There's one that's found mostly on mast cells and basophils, and this is responsible for the immediate hypersensitivity reactions we know. So this is the alpha beta gamma 2 tetramer. We also have the trimeric alpha gamma 2 isoform, and this one, there's less is known about this one, but we think it's involved in antigen presentation. We see it on Langerhans cells, not as important, so we'll focus on the tetramer. So in the tetramer, the alpha chain is responsible for the IgE binding. So that's going to bind with that glycan-containing portion on the heavy chain. It has a very high association constant, and at most, in most scenarios, this high-affinity receptor is going to be completely saturated by IgE. So they really like to associate with each other. Something I want to talk about is beta chain truncation. So, it has a beta chain as well as an alpha chain. And the beta chain is called MS4A2. And so you can have various truncations here. I won't go into all of them, but something I want to note is that if you have exon 3 that's missing, it would be called the MS4A2 truncated variant. And what this causes is that the receptor is going to localize to the nuclear membrane instead of the cytoplasmic membrane. And what does this result in? Well, we see this result in mast cell death. They get arrested in the G2 phase, and their proliferation goes down dramatically. So what's the point of all this? Well, the point that I'm trying to get across is that this receptor is not just an IgE receptor. It's actually a proliferation factor, a survival factor, and this could be really relevant if we think about potential therapeutics, right? So could we potentially artificially truncate this beta chain and have that alleviate some allergic symptoms? It's worth thinking about. So now let's talk about a little bit of how the IgE and its receptor are actually related. So we notice that if you take mice and you knock out their IgE antibodies, the density of the receptors are also going to go way down. And if you re-expose them to IgE, those receptors are going to go all the way back up. So what we sort of infer from this is that the IgE binding is going to heavily stabilize the receptor and prevent it from sort of retreating back into the cell. The way that it does this is it sort of decreases protease access to the receptor. So if the IgE isn't there, the receptor can much more easily be internalized or be degraded. And this is part of the reason why anti-IgE therapies like Xolair or the general name omalizumab work really well, is because they not only depleting what little free IgE there is, but over time this ends up drastically cutting down the amount of receptors there are in the first place. And that's going to help you reduce the threshold, increase the threshold of reactivity to a certain allergen. So if we're going to talk about the signaling pathways, the first step in that signaling pathway, and remember this is how we're activating a mast cell. So at the very end, this is sort of, we're going to be reaching mast cell activation. There's a lot of steps here. So I'm going to sort of show this, and you're welcome to read through these. This is actually already a truncated version from what is presented alongside this image, but I've cut it down to even its most key points here. So you're welcome to go back and read through what I've written, but these are the big steps that I want you to know. So the allergen binding to the receptor is going to just result in a signaling cascade. Again, if you're interested in all of those small steps, you're welcome to go look at them, but I don't think that they're critical to understanding the big picture here. And it basically forms a very complex signaling complex. And a key intermediate that I want to draw attention to is phospholipase C gamma. And so this molecule is going to catalyze the cleavage of this molecule PIP2, which yields the products IP3 and DAG. So really the big takeaway here is that the key molecules I want to focus on are IP3 and DAG, or diacylglycerol. And these two are going to be related to very directly to how the mast cell degranulates. So let's talk about that. So granule exocytosis, or degranulation, is actually very regulated by calcium. And once we know that, many of you might start to connect the dots, because DAG and IP3 are very related to calcium signaling. So IP3 and DAG, they each sort of do their own thing that is very important to how calcium, the high affinity receptor signaling, and degranulation are related. So DAG itself is going to lead quite directly to degranulation. And degranulation is going to result in a decrease in the calcium. So in order to bump up our calcium levels, we're going to have IP3 stimulate the endoplasmic reticulum to release internal stores of calcium, calcium ions specifically. And then once the endoplasmic reticulum stores are depleted, this protein called STIM1 is going to open calcium channels that will allow extracellular calcium to enter the cell and replete those stores. So really, degranulation is very directly related to calcium, as you can see in this image, right? So liberation of calcium from the endoplasmic reticulum is going to quite directly lead to degranulation. And that is also going to occur through DAG, which interacts with this intermediate molecule here. So let's talk a little bit about the low affinity receptor before we wrap up. So the low affinity receptor is also called CD23, and calling it the low affinity receptor is not entirely accurate because it actually binds quite strongly. It's just not as strong as the high affinity receptor. But it's one of the few immunoglobulin receptors that isn't in the IgE superfamily. And there's a lot less that's known about it, so I only have 2 slides on this. The first thing I want to say is that similar to the high affinity receptor, we see that its expression is quite correlated with IgE levels. So if you take away IgE levels, the receptor is not very stabilized, and it's going to downregulate, either getting degraded by proteases or just internalizing into the cell. Now, as far as the functions go, it is doing a few things. One thing that we see it do is as a transport protein. So it helps move IgE complexes and allergens throughout the GI and respiratory epithelium. So this is important because it basically provides a non-inflammatory pathway to sequester IgE. So if you look at this image here on the left, if the free IgE binds to high affinity receptors on a mast cell, this is going to result in all of the inflammatory pathways that we just talked about. It's going to result in degranulation. But if it binds to the low affinity receptor on a B cell, this is going to be called a non-inflammatory pathway. This is going to sequester some of that IgE away, and therefore we can say that CD23 is a sort of immunoregulatory protein. And we see this experimentally because when we knock it out, mice tend to have increased IgE production and more severe airway inflammation as well. So that sort of wraps it up for this lecture. I hope that provides a sort of overview of how IgE is structured, how it's working. And yeah, from here I would go and look at the lecture on histamine and antihistamines, because if you look at these granule components here, well, one of the biggest components there is going to be histamine.